So the motivation as to why we want to start developing and speeding up the development of our crops is in order to feed everyone in 2050 with this rapidly growing human population. The trend is in terms of wheat production, where the projected trend is not good enough to feed everyone, essentially, at the moment. So it sort of reflects the slow rate of genetic improvement of our crops globally. On this y-axis, you see this is the wheat yield in tons, and the green is where we need to be, and we can get there in terms of improving the efficiency of our breeding, as well as productivity on farm by pairing better genetics with better management practices. And if we can start to even fuse these things together as we go in the breeding process, I think we're going to really set ourselves up for even some pretty massive gains. So the future's not bleak, it's quite bright, I think, with all these new technologies coming online. The red line is actually where we could be if we stop breeding. So I think that's a good example of highlighting the importance of breeding and genetic improvement for things like rapidly evolving pests and diseases and climate change. It's very similar to Ben's visualization of a breeding pipeline, and we see it has 3 different phases: line development, where we're generating new inbreds, a field testing component, and the process which goes on to release that variety and make that seed available to farmers. But if we look at this line development phase, it can take up to 7 years. So if you're only doing basically 1 generation a year, and let's not lose track of the fact that a lot of people, a lot of breeding programs, a lot of crops are only doing 1 generation a year. In Australia, we're really fortunate. We've got some pretty advanced wheat breeding companies adopting a lot of these new tools and approaches, really pushing it in terms of the genetic gain, and they're doing 2 or 3 generations a year. Even with something like shuttle breeding, doing 2 generations a year is really good because it halves the time in which inbreds can be developed. So the field testing component is a tricky one because it's hard to get around this. It's needed to generate enough robust data across at least 3 years, if preferably more, to handle the G/E and to make sure that the variety that's being commercialized or delivered to farmers are actually going to perform on their farm. And so it's really hard to shrink that part of the breeding program. Of course, some of the new approaches in the prediction space that Ben mentioned with genomic prediction can shorten and get around this, but you still need to go down that path for variety release because you need to validate the performance of these lines in the field. And of course, the seed multiplication and release pipeline, it can take a lot longer than 3 years. If you're starting with a small amount of seed, it takes many generations to build up enough seed, and it can easily take a lot more. And that's where all the agronomy comes in. They start fine-tuning the enhanced genetics to find out what sort of management practices are going to be more effective for producing that variety. Again, some of the latest thinking is that if we can actually incorporate some of that information earlier in the breeding program and start developing genetics that are tailored to specific management practices, our colleague Mark Cooper, who's just joined UQ, is really into this, and I think this is a really promising space. So it's about, we have to basically choose, a plant breeder will have to choose parents to start the next cycle, and he or she needs this reliable information to do so in the absence of genomic prediction. So plant breeders have several different tools or strategies in which they can shortcut this whole process in terms of breeding cycle and getting this down, because as you saw in Ben's equations, the time in which and the length of the breeding cycle is a really critical part of that genetic gain equation. So one of them is the famous shuttle breeding, double haploid technology, genomic selection, and of course speed breeding. I'm going to talk about it today and explain a bit about this tool. But just to introduce these other tools, which are all very useful, and shuttle breeding has really been a revelation for wheat breeding and supercharged the green revolution, I would say, where Borlaug was acknowledged to be the one to sort of introduce and initiate this shuttle breeding program in CIMMYT in Mexico, essentially reducing the length of the breeding cycle by half because they grew 2 generations a year. And I can just imagine, like at the time where people were proposing to do shuttle breeding, it would have been something pretty radical, you know, like let's put all the wheat lines on the back of the truck and drive them up there or down there and let's grow them again. And the workers, I can imagine, wouldn't have liked that because it would have doubled the work. And it would have been really radical at the time to do this. But now shuttle breeding is really adopted by lots of breeding programs for lots of different crops. You take maize. A lot of the big breeding companies in the US have off-season nurseries in Hawaii, when it's too cold to grow maize in the US, they can grow it in Hawaii, and also is a nice place to go and collect an extra set of notes. You know, we're in Australia, wheat and barley breeders are using cooler parts of Australia in the south, where we can grow wheat and barley under irrigation over summer. So places like Tasmania are still quite cool in summertime, and coupled with the right irrigation, they can push through an extra generation a year in the field. Often they use this as a seed increase for the following season. So it's been a really valuable tool, and one other aspect of this shuttle breeding has been by applying selection in each of these 2 locations, it's actually enabled selection for broadly adapted wheats, and so which have been very successful globally through the CIMMYT network. And in 2008, there was a variation to the shuttle breeding introduced in CIMMYT, where they started incorporating selection for UG99 resistance in Kenya through the Kenya's rust screening nurseries. And because this was a huge priority with this evolution of a new rust strain, so you know, they come up with this 3-way system of testing and breeding in Mexico to introduce this enhanced rust resistance into their breeding materials. So this was also a very effective approach. Double haploid technology has certainly been has had a massive impact in plant breeding, particularly for crops like maize. It really fueling the hybrid testing programs. Essentially, it actually bypasses this whole process of single seed descent for inbreeding that's needed after a hybridization or cross. So normally, in my previous slide, I showed the process of single seed descent. Once we make a cross between 2 wheats, we produce an F1, of course, and then F2, F3, all the way to F6 as inbred lines, which can then be evaluated and tested across multiple locations. But double haploids, where you can make a cross and apply, say, maize pollen, which induces haploids, and in the lab, through a pretty intensive tissue culturing process and use of chemicals, they can essentially double the chromosomes. And once you raise those little plants up and get seed from those, they're often a bit shrivelly and not very good quality. Another generation is usually required to get enough seed. So there's a few generations still required for double haploids, even though you're making a cross and going straight to homozygous lines. It still takes time, and it can still take between 12 and 18 months, depending on the pipeline. For winter wheat, it probably takes a bit longer than spring wheat because of the actual physical time. You've got to grow the embryos, do this tissue culturing process in the lab, and produce the self-seed, etc. And when we weigh it up, certainly double haploids have been very useful when it comes to QTL mapping. It's very powerful because you get large blocks of DNA, improves the precision for mapping, or power rather, and of course, you get to that immediate homozygosity, but there are some disadvantages that I would like to talk about. And one is certainly that plant breeders, they love to do early generation selection in the F2, F3 stage when everything just segregates beautifully. And so you can't do that with double haploids. You get what you get, and that's it. It's sort of a random process, and certainly the double haploids work very well for close crosses, and that's how breeders have been using these very successfully. When you do wide crosses, you get a whole bunch of rubbish, essentially, and many of which are just discarded before actually field trials take place. Certainly you get less recombination. You get equivalent to the F2, and some would argue that that's a good thing because you keep these big blocks together, and probably in closely related material it is a good thing. But by going through the process of single seed descent all the way through to F4, F5, F6, we're getting additional recombination. And of course it can be quite laborious to set up your own in-house system for producing double haploids. A lot of companies and researchers outsource double haploids. In Australia, I think it still costs about $30 a double haploid line, and it can be quite expensive when you start to talk about big numbers for breeding. And so I had an interesting discussion at the last BGRI with Hans Brown about this over breakfast, and he was, you know, at the time they were weighing up whether to invest more in double haploids or go down the SSD path, and he was saying, well, what's the cost? I can't do the German accent very well, so I won't try. But he was saying, like, what's the cost of making 100,000 double haploids? And sure, it could be quite a big cost depending on how you do it, how you set it up, and what sort of service you use. Genomic selection, as we heard from Ben, it was a beautiful and simple explanation of how genomic prediction works. And you can follow up more with Ben this week. But just very briefly again, it's essentially shortcutting this testing of the selection candidates. So it's another visualization of a breeding program. I'll push too many. So basically, you're recombining the best individuals, generating selection candidates either through a double haploid program or SSD program, and normally they would go to the field for testing. But this you can genotype them and predict them, right? And so you can select your best parents, which go loop back into your breeding program, or they go on to variety release. And that's where they need to go through that field testing to actually validate performance, and that's needed for variety release. So I won't talk any more about that because Ben's covered this, and we're going to hear another talk about from Philemon later as well, about some examples on genomic prediction after lunch. But I'll talk a bit about my specialty in speed breeding. And I suppose we're initially inspired by NASA-funded research that was trying to grow wheat in outer space. The way that they were doing it was they were exposing the wheat plants to continuous light. So the plants didn't sleep. It actually tricks them into the reproductive phase, so producing grain very, very quickly, much faster than normal. And we thought what a cool tool that would be to maybe apply to our wheat breeding and research down on planet Earth, right? We started doing this back in 2003 with my colleagues at the University of Queensland. And when we first started doing this, we hadn't grown wheat plants before. So they looked terrible because we had the wrong, soil mix and the wrong fertilizer situation. But once we learned how to grow wheat and we got the temperature conditions right and we exposed the plants under continuous light. At that point, we're using sodium vapor lamps, as you can see in the picture. And what do you know? The plants actually grew really well and really healthy, and actually even resemble the plants in the field. And so once we refined this tool, we then use this for our day-to-day research and breeding over the last 6 to 7 years. And so we've been working closely with several breeding companies in Australia as well to exploit this tool. And so, of course, as Kai mentioned, there was quite a bit of media on this recently when we published our paper. This was one of the comics from the newspaper, in the West Australian newspaper, where she's growing her wheat populations in the Shuttle about to take off to to space. So everything okay. Yep. Counting down. I thought that was funny. And so I thought to start off, a lot of people are very skeptical about, how the plants grow and what they look like in speed breeding. And so here is a beautiful example of it's a time lapse of wheat growing. On the left, it's speed breeding versus normal breeding. The guys in the UK helped out with creating this time lapse, and because seeing is believing, right? So you gotta see it. So here we go, 4th leaf stage on the speed breeding. Elongation, there's the flag leaf going up on the speed breeding. Spike, flowering, and starting to senesce and die. And meanwhile, our normal breeding plants are just starting to elongate over here, and our wheat crop over here is in speed breeding is mature and done. And we couldn't go any further because they were just taking too long in a normal breeding. Normal breeding sucks. In a glass house. Okay. Oh no, we've already seen that one. Okay, so essentially what it means is we can go from seed to seed within 8 weeks. And there's variation depending on germplasm, of course. So for our spring types, a lot of the varieties and genetic backgrounds that we're interested from Australia and CIMMYT, most of the material are cycling in this time in our system. Here you can see a pot, because we use different configurations for growing our plants, cell systems, pots, and depending on the purpose and what we're trying to do, whether we're trying to cross. If we're crossing, we're using a pot system most of the time. And here you can see 4 plants growing in like a 1.6 litre pot, and they're producing on average about 3 or 4 tillers each, and they produce pretty nice spikes for crossing. And we routinely do all of our crossing in the speed breeding system. It's much easier, we find it easier. And we've made considerable numbers of crosses. So in the past, we've made 3,000 to 4,000 crosses in a year, just with a few people, for a recurrent selection program in the speed breeding system. So we can do large numbers of crosses, nowhere near as how many crosses that CIMMYT are making, of course, and some of our big breeding companies in Australia. But for our research purposes, we're doing okay. And here's the Suntop, a very popular wheat variety in Australia. This is 11 days post-anthesis. And so we actually harvest the grain 2 weeks after anthesis. Or we can, if we want to make the generation faster. And so this figure is a really nice example of the process and the options that we have. This was included in our Nature Plants paper from earlier this year. And so basically from anthesis time, we can harvest the spikes about 2 weeks after anthesis. They go into an air force dehydrator at 35 degrees for 3 or 4 days. And it's funny, the grains are green, yet when we bring them out from the oven, they turn brown. It's like a fast maturing process. But when they come out, they're not fully developed, and so often they can be a bit shriveled. Okay. Alternatively, we can go all the way through to maturity, and that takes a little over 2 weeks if we cut off the water about 2 weeks after anthesis. So normally it's about a month after anthesis if we start reducing the watering, and we can harvest the plants when they turn brown. And so how do we get the early harvest or the premature seeds to germinate? Was a really nice trick. It's using cold treatment. So if we're working with small numbers of lines, we use just Petri plates, but we can use cell systems and wheel them in and out of walk-in fridges and stuff. But here you can see we add the water, imbibe the seeds overnight at room temperature, and so they all swell up and they grow, and then they don't germinate very well because they've got like a fake dormancy that's been induced. Because they've been harvested early, there's an imbalance in the ABA in the grain, so the grain doesn't germinate. So what we do is we put them in the fridge for a few days, 3 or 4 days, pull them out, and hey presto, they all germinate, and we get pretty good germination rates of upwards of 90%. So if we apply this germination treatment. And what this means is that there's no need for embryo rescue, which is really cool, because that was a popular technique in the past, where people would harvest a couple of weeks after anthesis, but you'd have to go through this whole terrible tissue culturing process in the lab, and you can't handle large numbers, and you need all these specialized skills. But this gets around this, and we can handle literally hundreds of thousands of plants at once and treat them in a very rough and easy way, which is what we needed for plant breeding. And so, incidentally, speed breeding works for a whole bunch of other species, which is really cool. We work a lot on barley, so we also apply this to all of our barley breeding and research. And here you can see some beautiful comparison photos that Amy Watson captured in one of her experiments. And you can see on the left is speed breeding, and on the right is regular glasshouse or normal breeding. And these plants were sown at the same day and grown under the same temperature, but the only difference was light. So in speed breeding we had a 22-hour light, so we actually were nice to them. We gave them the 2-hour dark phase. And in the regular glasshouse it was 12 hours light, 12 hours dark. And so you can see that just the crazy difference between the growth rate of these plants. Chickpea, the flowers are there. Canola, it's a little bit slower for canola because it takes a bit longer to harvest the viable seed because they're so oily, and they take a bit longer to form. We learnt that the hard way. And yeah, and the list is growing. So some of our colleagues at UQ have been working with the peanut breeding company in Australia, and they now run their whole breeding program through speed breeding. So instead of achieving 2 generations a year, they do 3. And it's the most impactful thing that you can do in a breeding program. Just to achieve an extra generation a year, the genetic gain goes crazy. So you can tweak accuracy a little bit and do all these things, but I mean, that's a big impact. It's working for a whole bunch of other crops, Metacargo, Brachypodium, model species, which is really good because it can help speed up some of the research. And a lot of these figures and data that we have now to quantify the benefits of speed breeding has been thanks to some of this crew, Amy Watson, Shreya Ghosh from John Innes Centre, PhD student of Brandon Wolf. I believe they're around at this conference too, so please have a chat to them and ask them, if you've got more questions about speed breeding. But we teamed up with the guys at the John Innes Centre and the University of Sydney actually as well, had a similar protocol. They had developed a LED system in just a regular room like this with an office air conditioner. So it was a pretty low-cost setup that they used to speed up some of their rust screening and backcrossing work. And so we teamed up with all three to provide our protocols and make them available to everybody, because essentially we were getting sick of being harassed by emails because we got requests from more than 100 breeders and scientists from 24 countries, and obviously there was a demand, and the protocols really hadn't been published very well, and communicated very well. So we wanted to make this available to people. So yeah, there's a bit of a visual summarization of speed breeding and glasshouse with the light periods and the temperature controls. Essentially, we're using 22 degrees during the daytime hours and dropping the temperature a few degrees down to 17 when we give it the 2 hours dark phase. It just seems to help the plants recover a bit, and they're generally a bit more robust when we give them this little break. Yeah, but what it means is we can do up to 6 generations of wheat, barley, chickpea, and about 4 for canola under speed breeding conditions. And there is variation, so there are slower genotypes or varieties for each of these different crops. So it might mean that we can only do 4 for wheat in some backgrounds that have vernalization requirements. But I didn't mention, but of course we published this in Nature Plants, and it was great exposure, I think, for the tool, because it made the front cover of Nature Plants, and we got a lot of media out of this, which was great to help spread the word on speed breeding, and lots of people are trying it and giving it a go now, which is great. Here's Carl Schmidt. He's got a system working on speed breeding for amaranth and quinoa. And here's a photo of our recent experiment in lentils, working with the University of Melbourne. And the lentils is really cool. Like this little guy here on the right is a land race of a lentil that normally takes 160, 170 days to flower under regular glasshouse conditions. But we give it speed breeding and it flowers in 27 days. So we're currently trialing all the relatives of lentil under speed breeding that just take forever to flower, and hopefully it'll open up a whole new gene pools in terms of improving some of these crops. According to Twitter, lots of people are trying speed breeding now. We have an effort, a really nice system set up in Turkey, where they're actually backcrossing some quality traits into some of their wheats using a LED indoor system. There's a DIY effort in California where I only know her name by the Twitter handle, Chaotic Genetics, because she works on peppers, and she's actually sown this speed breeding unit out of materials, reflective materials, set up LEDs inside that are computer programmed, and she's actually using this to accelerate some of her research. And we have speed breeding in Minnesota working well for oats, and apparently the oats were getting through in about 40 days. Barley was faster. Plants off to New Zealand sooner this year. So we hadn't actually tested oats in this system, but it sounds like they're working quite well. There's some activities going on led by Susanna at CIMMYT to trial speed breeding. And this initially started off in the main headquarters and trialing and looking at how this system would work. But there are future plans to scale up or make a larger facility in Toluca. And pending on the success and how this thing works, they might expand it into other activities like rapid cycle genomic selection. But certainly I think it would be quite promising for marker-assisted backcrossing and speeding up some of these pre-breeding efforts, that's for sure. And Susanna's going to talk tomorrow or today? Today, after lunch. So Susanna will be around, and please approach her. But speed breeding also has some nice applications to speed up the research, particularly around transgenics and CRISPR pipelines. And in many countries, the good news is that CRISPR is appearing to be accepted as a non-GM way of improving our crops when we're inducing mutations. So I think this is really exciting. But essentially these are, you're asking how can speed breeding do this? Well, here's some calli transformed of golden promise that were actually produced in the speed breeding system. And then they were grown on under regular glasshouse conditions and speed breeding, and you can just see the time savings in which you can actually just physically grow those plants. So what it means is that we can deliver these new traits into farmers' fields faster and speed up this whole pipeline, because it's just about producing plants and growing them quickly, right? And getting those seeds. And it also means, well, from a research perspective, you can publish that Nature paper faster too. So for some people, that's important, But it means that we can halve the time for validation of these sort of studies. It's going to speed up, the research that feeds into all these breeding programs too. Yeah. Here's a photo of some of our near-isogenic lines of barley. Sorry, it's not wheat. But we're doing it for wheat too. It's the only photo I had of the nils. These are BC4 derived nils on the last generation. Took about 18 months to create. And essentially we were tracking different variants at the MLO locus and transferring them into this Bodan background. Bodan is extremely susceptible to powdery mildew, and the guys at Curtin University Simon Elwood, he suspects that some of these new variants don't have trade-offs in terms of being heat sensitive or to abiotic stress. And so, these become really useful tools, like to actually validate these new alleles, because these are going to be seed increase this year, and then they'll do extensive testing for yield in different environment types to look at the trade-offs for these different variants. So we can create neo-isogenic lines really quickly, and some of my crew are doing this for a whole bunch of alleles, from the Vavilov wheats for stripe rust, stem rust, to yellow spot. And while they're trying to track down the genes through mapping and creating new populations, because it helps us do this too. So we start off with a massive collection. They create a very targeted bioparental population to map and fine map those genes. But in parallel, we're actually creating backcross-derived lines in the speed breeding system, where we're feeding in those new alleles into those adapted backgrounds, which we then deliver to the breeders. Normally this takes a long time, so speed breeding can help speed it up. Speed breeding 2.0, I copied it from Ed Buckler. Ed Buckler's always talking about breeding 1.0, 2.0, 3.0, 4.0. 4.0 is where everything just goes crazy, And but speed breeding 2.0, it's all about reducing the costs of operation, so we can actually do this on a large scale, and ramping up speed breeding and getting serious for commercial breeding. And so we've just replaced all of our lighting at UQ with these new LED lamps. It's actually they're made by a company called Heliospectra. There's a few different options on the market. Our Glasshouse service has just tested a few different brands, and this one turned out to be the best. I don't have shares in Heliospectra. I'm sure the other companies are just as good. But incidentally, the guys that joined in this centre also chose Heliospectra. So seems like it could be just a coincidence, but it seems like they're doing a good job. The plants actually, I think, are looking even healthier under these lights, because they're only giving the plants the red and the blue spectrum that the plants need to actually grow. So way better, way more efficient than these sodium vapor lamps that we had in speed breeding 1.0. And so that means that they're cheaper to run in terms of electricity, but they're not producing extra heat load that is required to either cool or in summertime for us. We would normally be then using more electricity to cool that environment down. And so I couldn't help myself. Here's a picture that National Geographic has chosen to run in the next issue, which is pretty cool. And it's all about using LEDs and for boosting plant breeding. And one of the things I think when we think about how we can use this tool more broadly for food production, I think there's some nice opportunities to integrate speed breeding approaches into indoor farming systems, and it might mean that we need to tailor the actual varieties that we grow in these systems to be productive and be faster generation cycling under speed breeding conditions. Good example. Do I have time? Let me see. Maybe. A good example is tomatoes. Tomatoes were sensitive to continuous light. So they go all necrotic and basically die. They don't like it. But researchers in the Netherlands, they cloned a gene which makes the tomato plant tolerate these continuous light conditions. They plugged that gene into a variety, grew it in their glasshouse under continuous light conditions. And let's don't forget, like, the Netherlands are the #1 exporter of tomatoes. It's crazy. They got tomatoes as far as the eye can see. And what this gene did by growing the tomato plants under this continuous light, it boosted production by 20% productivity of tomatoes. So I'm thinking that we can do this probably for a whole bunch of other species which we grow indoors, or can open up new opportunities in way we grow food. In the field for a lot of crops, it's really frustrating because there's dirt and you've got to wash all the produce. But indoors, you don't have to spray them. It's clean and you don't have dirt. So there's a lot of opportunities, I think. Even Elon Musk's brother has started a venture in New York where they're growing. This is actually a picture of one of his shipping containers. They got decked out where the crops are growing in a vertical library So I think there's a lot of innovation going on in this space and a lot of new things coming online, and we're excited about LEDs. Well, there's new technology with laser lights, which is mind-blowing. So the lasers are only giving specific wavelengths, and you can have these laser lights outside of the growing conditions, and they go through to the plants, and so the heat is outside and removed of the growing environment. So the costs of running are even cheaper for maintaining temperature. And I guess one of the key aspects of speed breeding 2.0 is scaling up for commercial breeding. And here you can see barley and wheat growing under different densities in cell systems. And Amy Watson did these beautiful experiments where she's measured all these different traits and documenting the changes and behavior under different densities, just as like you would do in the field, I guess, with agronomy trials. And so we have 900 plants per square meter, 576 and 270, and what do you know? There's a pretty high rate of survival in the 900 plants per square meter, and which is really ideal for breeding. We do see changes in behavior like decreases in plant height. At high density, you can see they're much shorter overall, and we do see nutrient stress when they're growing under high density, and this is great because it pushes them to go a little faster. So we're not too worried about this yellowing and the lower leaves in the canopy. And when we look at the 900 plants per square meter, they're producing 7-8 seeds still, even when they're growing in that density. And that's all we need. We don't need, actually, that's too many seeds. We don't need that many for SSD. We only need one to go to the next generation, right? So it depends again on what sort of purpose you're using the system for. If you're just creating inbreds from a cross, then you can grow them at this density and start with a larger population size than what you need, because you lose a few along the way. And of course the larger spikes at lower density. And when we look at the plant breeders equation, of course all this speed breeding work is coming in the years per cycle, where shuttle breeding was coming in, speed breeding is coming in, and helping to accelerate genetic gain. In terms of the breeding pipeline, again, if we go back to this figure, where it's all about shrinking this line development phase, and it really can do this if you're talking about generating inbreds within 12 months or 18 months. So I think it has a lot of potential. If we look at the breeders equation again, we can start to perhaps use speed breeding for selection for some simple traits as well. And we've done that. Actually, my honours project about 10 years ago was working on grain dormancy. And we developed some phenotyping methodology to actually select for grain dormancy in the speed breeding system. Unfortunately, it slows you down a bit because you have to harvest the grain at full maturity. You can't do the early harvest to test for grain dormancy. So it means we can do about 4 generations a year. And over the course of the last 10 years, we actually introduced grain dormancy, a high degree of grain dormancy, controlled by a large number of genes. We integrated it into several different backgrounds, but one of them in the E.G.A. Gregory background for Australia has just been released in partnership with Dow AgroSciences. And so this represents the first variety bred completely under speed breeding conditions. Dow took over the field testing and quality testing component. And why this is a big deal is because we don't actually have any PHS or pre-harvest sprouting tolerant wheat varieties in Australia. They're all white grain wheats. They're generally pretty high quality, and they don't have grain dormancy. Grain dormancy is much more frequent in red grain material. So, and researchers have been trying to solve this problem for a long time. And so without something like speed breeding to get through all those generations, I think we did something like 20 generations to breed this variety. It just would have never have happened. We would have given up, or it would still be going. So it took 10 years to get this variety on the market, but it was a totally new trait, and lots of crossing and a lot of selection involved. We got triple rust screening. How am I doing for time?
No, good.
It's okay. We also, well, when we first started this work, we thought, what other traits can we do in speed breeding. And of course, there's pre-existing rust screens that you can do in a regular glasshouse. So we thought, oh well, maybe if we tweak those and we could get them working in the speed breeding system, because wouldn't it be nice to screen our populations all year round and for adult plant resistance, and use this as a tool for introgression. And so we started doing this. We just launched straight into it, really, from a breeding perspective. We just started layering on the rust. We developed a nice system where we layer on the 3 rust as the plants grow. So we put up stripe rust on first, and then leaf and stem rust. And what happens is, if we get the timing right, all 3 rusts develop at once, and it's beautiful. But what it means is we can sort the plants at once, one time only, and just select the green ones. And we're assessing the stripe rust on leaves 3 and 4, looking for chlorosis and necrosis, and stem rust on the stems, elongated stems, and leaf rust on the upper leaves, so the flag and flag -1. We're looking for those reactions. And so this became quite a useful tool for us, working with breeding companies. So we've been working with one company now for 6 or 7 years using this screening approach. And they were so impressed with the material coming out of this pipeline that they increased their investment in this approach. So, well, not de-invest totally, but started to invest less in double haploids. That's because we're generating material that are highly selected for fixing some of these key traits in their materials, and they don't have to worry about those traits so much when they get to the field. So it's not perfect. Some susceptible ones still get through, but as long as we're right more often than we're wrong, we go forward, and we're really enriching these populations with the right alleles. And so the cool thing is, when I got the opportunity to start my own team, we had a whole bunch of fabulous PhD students, and we could go back and do the science. And because we were doing it from a breeding perspective before, we were doing it pretty dirty. And so Adnan set up some beautiful experiments where he tested different standards for rust resistance in different environments in the field, and then speed breeding, looking at correlations, making sure that these traits were correlated, of course. We've done the same thing for tan spot resistance, and Eric has done a lot of this work. One of the traits we're interested in tan spot and exploring is adult plant resistance, which hasn't been well explored and understood at the moment. And so he's developed a nice system where we can infect the seedling plants, evaluate those, and grow on the plants under speed breeding and test the adult response. And we've identified some really nice new alleles that are giving us adult plant resistance, even in the presence of this major TSM1 tox-A interaction that's going on. So it seems to be mediating or reducing these effects. So this is something that we're exploring. Other traits that we can phenotype in the speed breeding system is some of the physiological traits. This is a figure from our paper in Nature Plants, and work was done at the John Innes Center, and you can see, so orns, RHT, loci, wheats carrying different combinations, giving different phenotypes, and FHB resistance can also be phenotyped in the speed breeding system. And I believe the guys in Canada, some team in Canada have also developed a similar system in control conditions for FHB screening as well. Is that right? Maybe.
Maybe just a question. Are you talking about spring wheat, about everything? Because for winter wheat, is it a little bit different, probably?
That's right. I'm talking about spring wheat, and winter wheats still need the vernalization treatment before they go into the speed breeding system. And it's much better to still grow them in the speed breeding system than a regular glasshouse once they've been vernalized. Yeah. So unfortunately, we haven't solved the vernalization issue yet, but I know there are lots of people around the world that are working on it and trying to basically silence the key flowering time genes with anti-GM, or so not anti-GM, non-GM approaches. Yeah. There's lots of anti-GM people on Twitter. Yeah. So
just a question, quick question. So did you have a chance to just compare your results, like the phenotypic results for the APR rust genes, when you are phenotyping your population in the greenhouse compared to the same population when you are phenotyping in the field? So most of the rust people, they say that probably you can't get the exact results when you are phenotyping for the APR in the
Yeah.
greenhouse. So did you have any chance to look at that how your severity actually changed when you are phenotyping in the greenhouse compared to in the field?
Yeah, there's so the tricky thing about adult plant resistance is that, as you know, there's probably a lot of pathologists here that are familiar with this, that these APRs are just interacting quite a lot with the environmental conditions. You know, warmer temperatures, cooler temperatures, and you get different expression. LR34, RPH20 in barley. Cooler temperatures are generally better for enhanced expression. And so we see this variation in phenotypes across environments. So when we take a set of phenotypes from one environment and we look at the correlation with another environment, it's never perfect. It's never 1. And sometimes it can be as low as 0.6, or right? So what we've done is we've worked with larger population sizes, around 300 or so, and we get correlations of about 0.8, you know, with some nurseries. And then sometimes it's lower. But the point is, we're using it as a tool, really. And if you're doing it from a research perspective, you do have the flexibility of actually running different temperatures or trialing this. And I think this can really help speed up some of the research side of things. But if you're just screening from a breeding perspective, you know you're not going to be 100%, and you still got to do the characterization as you go down the testing path. So these breeders are still doing this. Even the companies that we're working with, they're still putting the materials that come out of our pipeline, they're still putting them into the rust nurseries, of course, because you still get the odd line that's susceptible, you know. And so another trait my group is really interested in is roots. And Cecile Richard developed a nice system that's totally compatible with the speed breeding system. It's the clear pot method, transparent garden pot. It's not rocket science. It's literally a pot. And we sow the seeds around the outside, measure the root angle with an iPhone, and we can calculate this root growth angle, which is really important across. It's been proven now across a whole bunch, most of the cereals basically, that this root growth angle is reflecting the distribution of roots and important for water uptake and extraction, particularly at depth, where shallow root systems don't have many roots at depth compared to the narrow, deep, steep root systems. And so in our part of the world, in Australia, in the eastern wheat belt, we have deep soils and not much rain in wintertime. Most of our rain comes in summertime. So the crop's growing on stored soil moisture, and so we're trying to select wheats that have a deeper root system. And we can do this at the seedling stage. It's a proxy. Again, we're not 100% correct. There's other variation, but it's highly correlated with this adult performance. And what Cecile did is pretty cool. She performed the first direct selection for root architecture in wheat, maybe in any crop. I don't know. But what she's done is she made a whole series of crosses. There's a few crosses. Here's just one example. In a backcross background, she applied selection, bidirectional selection. So actually wide roots and narrow roots, and did this over 4 cycles in the BC1, F2, F3, and F4, and successfully changed the population distribution of those lines. And so we can use this as a tool to enrich populations quickly with desirable alleles for root architecture, and do all this in the speed breeding system 6 generations a year. So we're really excited because this develops some really cool materials that we can actually, you know, validate some of these things in the field. So now we're increasing the seed, and we'll do all these trials in different environments, but we needed the genetic materials to actually validate some of these traits. Samir, one of my other PhD students, is in the crowd. He's developed a nice system that integrates the clear pot with crown rot screening. So he's culturing this crown rot stuff in the lab on plates and transplants it on the surface of the pot. It just takes over basically and just infects all the plants. And he used a weighted selection index to select plants that had the desirable phenotypes for root angle, crown rot tolerance, leaf rust, plant height. And so using this index, he's also shifted the population really, really nicely for these multiple traits. And all in the speed breeding system. In terms of just quickly an example of a trait integration study that we did, this was actually part of my PhD work, and we wanted to demonstrate what speed breeding could do in terms of transferring some traits quickly. So we picked on this wheat variety called H45. It was high yielding, it was preferred by industry, but what happened, unfortunately, is a new incursion of, I'm not sure if it was an incursion or not, but it was certainly a new pathotype of stripe rust, I think YR7. I'm not sure whether that evolved in Australia or it came in. I can't remember. And so what we did is we took this H45 variety and used a modified backcrossing strategy with trait selection along the way to rapidly produce backcross-derived lines that carried triple rust resistance. So we applied triple rust screening after we made the three-way crosses. We produced a very large segregating population. We then did the multi-trait screening. Then once we found the resistant ones, we actually, because they were screened at the adult stage, we chopped them back, we transplant them into slightly cooler night temperatures, add a bit of fertilizer, and they regrow. We actually make the back crosses, because they were the special plants. When they go to the next generation, all the genes segregate again. So we wanted to capture them in that generation. Next generation, we did intercrossing to combine favorable alleles across the different lines, and then created another big segregating population, repeated our multi-trait selection screening procedure twice, and that increased the homozygosity of the lines that then entered the field for seed increase, and then the field testing component started thereafter. But this photo was taken in, I think it was 2015 or so, where we had some of the field trials in I think it was southern New South Wales at a farmer's day. There was more than 300 farmers there, and they were super excited about this variety because they knew it looked like H45, it smelt like H45, but it had the rust resistance. And so, some people don't like backcross breeding because you're sort of going backwards all the time and introducing traits, but it's a good example of how we can use the speed breeding system to integrate traits, and particularly from a pre-breeding perspective, it's a very useful tool. And I mean, it reminds me, this photo, of like because I loved fishing, right? That was my fun fact. And when you go fishing, you hold up your big fish. You're very proud of it. I was very proud of these lines, of course. And so, working with some of the breeding companies, we perform this multi-trait selection, and they sort of go through a funnel like this. It's an example of what we can do in the system. We can start off with grain traits because we can handle large numbers. So grain dormancy, tan spot, rust, plant height, maturity. We can throw out the double dwarfs. We can do all that sort of stuff, and end up with a highly selected set of individuals that can be genotyped, backcrossed, intercrossed. The sky's the limit in terms of what you want to do, what you can do. And so, as I mentioned before, some of my crew are working on the Vavilov collection, and for different traits, mining this collection for new alleles, in which we're backcrossing those alleles into adapted materials very quickly. So, and just quickly, I couldn't help myself but mention Speed Breed GS, which is a fusion of speed breeding and genomic selection, which Ben mentioned very briefly. We are doing this in practice, actually. Our PhD student, Amy Watson's developed some real populations, real breeding populations, where she's actually implemented Speed Breed GS and measuring actually realized gain from doing this. But we actually did a simulation study with the help of Eva in Huttersleipzig University in Germany, and the simulations were built on real data sets, real wheat breeding data sets from CIMMYT, provided by Susanna, and we essentially simulated 4 different scenarios where we had the phenotypic selection, which was very similar to a very standard breeding procedure. I think there was 2 generations a year using shuttle breeding. Genomic selection, which was just shortcutting this whole field testing process, and in order to predict the best parents for the crossing cycle, and Speed GS, which is actually doing 6 generations a year, and the genomic prediction to get around the field testing, and SpeedGS plus diversity, which was an element of pre-breeding, injecting new desirable alleles into the breeding program along the way. I'm not going to go into detail into the programs because they're super hectic, but here's the results, and you can see quite simply that the speed breed GS is a lot better than the conventional breeding because you're getting the double whammy. You're getting the double whammy of time from both the prediction and avoiding the field testing and 6 generations a year instead of 2. So it's really simple stuff, and you don't have to be a genius to work out that it becomes more efficient. But it was really nice to see with some of the simulation results, and we're talking in about 30 years' time, extra 34% genetic gain, and Kai did the sums, and it worked out that this extra 0.54 tons per hectare equated to more than 1,000 loaves of bread. So we talk about feeding the world in 2050, and so we thought this conversion was relevant, and look, this highlights what we need to be doing to speed up some of our breeding strategies. But incidentally, the pre-breeding strategy, you can see, continues the gain. There's a lot of debate whether that plateau you see here is actually an artifact of the simulation assumptions, which is probably true. If you talk to someone like Mark Cooper, who comes from Pioneer, he will tell you that plateau will never happen. So that's good to know. So in summary, just some of the future opportunities and challenges. Commercial breeding companies in Australia are either speed breeding with us or set up their own in-house systems, which is great. And that's the thing. They either need to invest in new infrastructure, and the big companies have done that, or outsource the service. And so I'm not really aware of many services yet, but hopefully, I'm anticipating that there will be some popping up. And I know that a lot of people around the world are developing their own infrastructure and systems. So there's a lot of grants going in Australia to build new facilities. It sparked a lot of interest. Our speed breeding paper, of course. One of the challenges is the energy costs of actually doing all this, and the same costs that sort of Elon Musk face. But the goal is to take this whole system off the grid with the new solar panel technology. There's even glass that they've designed for glass houses that captures some wavelengths of light for energy production, but let others through for plant growth. So there's crazy things happening. There's laser lights. Battery capacity is dramatically being enhanced. So I think it's very promising where all this is going. And the costs, of course, are a critical thing from a commercial breeding perspective. What other crops could benefit? Well, I mentioned a few, but there's probably some other ones that we can try this system for. Speedbreed GS, there's going to be I suspect, a lot bigger gains possible through redesign of the breeding program. With any of these sort of tools and technologies, it's very hard to just directly apply them and integrate them in a breeding program. It requires pretty drastic change and rethinking of the whole system. So, for instance, it may not be the best approach to wait until F6 in the speed breeding system to apply GS. Maybe you could do it earlier with the right models and everything. And of course, we could couple this with some disease screening and different approaches, different models, all the GS experts are working on that. So I think there's a super exciting opportunity to combine speed breeding and speed GS with CRISPR, and it's something that we're exploring to reverse alleles in parents' use for the next cycle in the crossing block. And I'm working with Ian Godwin at UQ, coming up with some pretty crazy ways in which we're integrating speed breeding with CRISPR and taking CRISPR out of the lab. And so I think this is an exciting space, combining these tools. And speed breeding is just one of them. So how can we combine it with all these other things? And some of my crew, thanks a lot.